17, that's the ending for the chapter. Don't forget, make a note, make a reminder something, because those are easy points to miss otherwise, so get that in. All right, and today we start chapter 17. Chapter 17 and chapter 18 sort of dovetail, and that's because it's the cardiovascular system. So we're going to start out, we're going to talk about the blood, that's today. Then we're going to talk about the blood vessels. And then in the next chapter, we're going to talk about the heart. And then the second half of the next chapter, we sort of put it all together and we're going to talk about cardiovascular system homeostasis, which gets sort of complex. But once we've got all the pieces, then we can sort of start looking at the whole thing together. So chapters 17 and 18 is sort of these pieces. We've got the heart, the blood vessels, that's arteries, veins, and the capillaries in between, right? And then you've got the blood itself, which is what we're going to spend most of today talking about. All right, so first up is blood. So what does blood do? In most of these organ system chapters, there'll be a table like this or a slide like this where we talk about the functions of this system or the functions of this organ. And the reason for that is none of our organ systems do just one thing. You know, they all have a primary function, but then they have other things that they do as well. The body is sort of amazing like that in that, instead of having one job, all of our organs really have multiple jobs that sort of fit together into one, big picture. So probably the primary function of blood is this first one, and that's transportation. The reason that we have blood vessels, and a heart that moves the blood around all the time is to transport stuff from point A to point B. For example, oxygen, there's lots of it in the lungs, but all of the tissues, all the cells in the body need it. So the blood goes through the lungs and then out to the tissues, carrying oxygen along with it. And we see that same pattern for all of the different organ systems. The kidney, its job is to get rid of metabolic waste products. Well, in order to do that, those waste products have to get there. How do they get there? Blood. So transportation of dissolved gases, nutrients, hormones, and metabolic wastes. So the gases we're going to talk about in the respiratory chapter. That's oxygen coming in and CO2 going out. Nutrients we're going to talk about in the 2 digestive system chapters. We're going to talk about carbohydrates, proteins, and fats. Hormones we've already talked about, right? We talked about that last week. And then metabolic wastes we'll talk about when we get to the renal system, when we talk about the kidney, right? So all of these things take place or are carried in the blood. And we're going to talk about how those things are carried a little bit today. And then there'll be more on that later. Okay, so that's the primary function. But what else does blood do? pH regulation. One of the themes that you'll hear a couple of different times in this semester is the body is very sensitive to pH changes. The proteins that make our cells do what they do and function correctly only do so at a very narrow pH range. So the body defends a pH of 7.4. And usually in the healthy normal person, it doesn't vary from that by more than 0.05. So it's a very narrow range. And anytime your pH is not 7.4, you're sick, at least a little. So patients in intensive care units and things like that, a pH abnormality will get you there very quickly because the body just does not tolerate changes in pH. So if it doesn't, there has to be ways to control that pH to keep it normal. And one of the principal systems that does this is found in the blood, and that's called a buffer system. A buffer is a chemical that, when present, stabilizes pH. So for example, if you've got a beaker of distilled water, right, and you add hydrochloric acid to it, right, the pH is going to drop, right, because hydrochloric acid is a very strong acid. But if instead you have a buffer in that water and you add the hydrochloric acid, the pH won't change until all the buffer is used up. So the blood has a number of these important chemicals called buffers. Now, we're not going to talk about that much in this chapter, but when we get to the acid-base chapter, you're going to see the role of blood in maintenance of pH balance. Okay? And then ion composition. So sodium and potassium and calcium are critical, their levels are critical for how the body does things. So the blood plays a role in keeping those levels where they should be. In other words, sodium levels high, potassium levels low, calcium levels within that normal range that we talked about in the last chapter. All right, so regulation of pH and ion composition. Restriction of fluid losses at injury sites. All right, this is where blood is far superior to water. You know, if you've got water in a plumbing system and you spring a leak, well, the water just keeps leaking and leaking and leaking, right? So you call a plumber who's going to replace the pipe. Our blood vessels, that's not the case. If blood starts to leak out from a hole, that hole, first it seals, and then that hole heals. So we have a multi-step process that essentially keeps the blood in the vessels where it belongs. And that's the clotting process, or the coagulation process. And we're going to talk about the multiple steps of that either today or next time. So it's an active system that can prevent blood loss. All right. Defense. The blood vessels and the blood contain our defender cells. You know, the immune system works because there are cells that attack things that don't belong. And those are the white blood cells. So we're going to talk about the 5 different kinds of white blood cells today. And their job is defense, you know, to keep that which does not belong in the body out. And if there's a breach of that, in other words, if a bacteria or a virus does gain entry, the white blood cells are going to attack that until it's gone, destroyed, you know, and back to being sterile on the inside again. Here again, we're going to talk about the immune system in the chapter after next, when we talk about the lymphoid system. But we are going to introduce the players today, the white blood cells. And then finally, stabilization of body temperature. As you know, we're exothermic organisms, which means we're warm-blooded. In other words, we create more heat than we need, and usually our body temperature is higher than our surrounding temperature. You know, the only time that isn't true is on very hot summer days, where you get above 98.6. That's a pretty rare occurrence. But we're warm-blooded, which means that all of the cells of the body are producing heat all the time. We have to get rid of that heat. You know, the human is fairly well insulated. We've got a layer of fat underneath our skin that helps to keep our body temperature in. But we still have to get rid of excess heat. So the way the blood and blood vessels do this is as a kind of radiator. You know, so if you are exercising, for example, what the body does is it pushes blood out onto your skin, or not onto your skin, but just underneath the skin. And as your heart pumps that blood around, the heat is radiated from the skin out into the atmosphere. So just like the radiator on your car is there to get rid of the engine's heat, the skin and blood work together to do that same thing, to get rid of excess heat by bringing hot blood just underneath the skin so that that heat can radiate out into the atmosphere. So, and then the reverse is also true. When it's cold outside, we can conserve our heat by doing just the opposite, by running blood away from the skin and down deep next to the bones. We can hold on to precious heat all by shifting the flow of blood. And we're going to see, we'll talk more about that when we talk about how the body uses energy in that metabolism and energetics chapter. But it is a key and critical aspect. You know, if it weren't for this radiator system, we would just keep getting hotter and hotter and hotter, and eventually we would die of heat stroke. So it works very, very well. All right, so blood, what's it all about? Well, it is about 40% cells and 60% water. Technically, blood is a fluid connective tissue. I know we haven't talked about connective tissue in a long time, but last semester we learned that connective tissues have a lot more stuff present than cells present. You know, like an epithelial tissue or a muscle tissue or a nerve tissue, it's all cells. When you look at it under the microscope, it's all cells. In a connective tissue, there's a few cells and a lot of other stuff, right? So blood is just like that. Under the microscope, most of what you see is not cells. So about 60% of what you see is what we call plasma. Plasma is the component of blood that is not inside a cell membrane, right? So plasma is whole blood with the cells removed, all right? So whole blood, or just plain blood, when we talk about blood, we're talking about this mixture of plasma and what we call the formed elements. Now, why do we call them formed elements and not just cells? Because one of the formed elements, the platelets, are not actually cells. They're pieces of cells. So we can't call it plasma and blood cells. We have to call it plasma and formed elements because that includes the red cells, the white cells, and the platelets are all in that formed elements piece, right? So we're going to talk about the plasma first, and then we're going to talk about the formed elements, and we'll talk about each of the pieces as we go. Some general properties of whole blood as it's circulating. Typically, its temperature is about 38 degrees, which is 100.4 degrees Fahrenheit. Now that doesn't match our standard body temperature of 37. That's that radiator effect. Typically, we have heat to get rid of. So the blood is a little warmer than the rest of the body because it's going to carry that heat away and then go and get rid of it at the skin surface. So it's slightly above normal body temperature. Blood is quite viscous. All right, well, what does that word mean? Viscosity is how thick or syrupy, I guess you could say, a fluid is. Technically, it's how much does a fluid resist motion. So the best way to look at this is as an example. You know, if you take rubbing alcohol, it flows very easily, even more so than water. You know, it flows very quickly out of a container. Whereas if you look at like corn syrup, it flows very slowly out of a container. So corn syrup is very viscous, and rubbing alcohol is not very viscous. So blood is about 5 times as viscous as water, which gives it, there's some thickness to it as it flows around the body. And that viscosity is quite stable. You know, in other words, the body keeps it at a very regular level so that the blood can flow easily enough but still do all of its many jobs. All right. And then finally, the blood is slightly alkaline. So 7.4 is the pH of most body fluids, and the blood is no different. So it's usually between 7.35 and 7.45. So a little on the north end of just neutral 7. All right, so first up is the plasma. So, let me back up. So it's about 92% water. Okay, that's the boring bit. But the other 8% is where the interesting parts are. So 7% is plasma proteins, and then 1% is other solutes. In other words, things like ions. All right, so let's look at the plasma proteins first. The plasma proteins, what are they? Well, proteins are long strings of amino acids, right? They have a particular shape. When they're dissolved in water and circulating in the blood, we call them plasma proteins. So 72% of what you find in, 7% of what you find in plasma are these plasma proteins. So some examples. Albumins. Albumins are proteins that are made by the liver. Their only job is to exist and to some extent to carry things around. They're produced by the liver. They're found in the blood. And what they do is, by being present in the plasma, albumins, as well as these other plasma proteins, they create an osmotic pressure. You know, anytime you've got a ton of stuff dissolved in a fluid, that fluid has a tendency to pull water towards it. And one of the ways that the cardiovascular system keeps water in the vessels, which is where we want it in terms of circulation, is to use osmotic pressure to make that happen. So the liver makes albumins, albumins pull on water, water enters the cardiovascular system and has a tendency to stay there because of all these albumins that are present. So we're going to talk about osmotic pressure quite a bit when we get to the capillary. It won't be today, probably the time after next. And you'll see what I mean about this, how these albumins and these plasma proteins pull on water and help to keep water in the vessels where they belong. Okay, so that's one big class. Globulins, they're called globulins because they're glob-like. In other words, if you look at them chemically, if you look at their structure, they don't really look like anything. They look like globs, so they got called globulins. They also have a tendency to sort of stick to each other. But globulins are another big class of proteins, and they can do many different things depending on which globulin you're talking about. So like antibodies, which we'll talk about in the immune system, they're an example of this group. There are transporters, like hemoglobin is a globulin. Now, hemoglobin is usually found inside of cells, but globulins can move things around because they can hold on to chemicals and then help them move around the cardiovascular system. All right, so albumins, globulins. And then fibrinogen. Fibrinogen is about 4% of the plasma proteins, globulins 35%, albumin 60%. But fibrinogen is a key and critical part of the coagulation system. So fibrinogen is present all the time, and it's soluble. In other words, it stays dissolved in the blood, in the water of the plasma. However, when fibrinogen is split to make fibrin, fibrin comes out of solution and helps to make a blood clot. So without fibrinogen being present, our blood doesn't clot, as we'll see when we talk about the clotting system later on in this chapter. All right? And then, so these are the major players. So 60%, 35%, 4%. And then there's a bunch of others, but they're found in much, much smaller quantities, right? So in the plasma, there are proteins doing all different kinds of things. You know, there's enzymes catalyzing chemical reactions. There's transporters. You know, there's all kinds of different roles there. All right. So that's 7% of the 100%. 92% is water, right? Well, the 1% is other solutes. And typically what we mean by this is non-organics. Proteins are organic molecules, and usually they're great big huge organic molecules. Well, this remaining 1% are very, very small molecules, even ions, single atom ions. So these are things like sodium, potassium, calcium, magnesium, chloride, bicarbonate, phosphate, sulfate. All of these, you'll see there aren't carbons in any of these except that one. These are critical for the body to function normally, like we learned last semester. You know, how many times do we talk about sodium and potassium, right? Well, without those being present in the blood, they're not going to be present in the cells or in the interstitial fluid where they have to do their job. So even though it's only 1%, these things are critically important, right? Even though they're tiny. You know, compared to a protein, a sodium ion is, a grain of sand next to a giant mountain. I mean, you have a huge size difference. But because these molecules are so small, they can be very active. So that's the electrolytes. Other solutes, the organic nutrients and the organic wastes. So things like fatty acids, small amino acids, carbohydrates, those are found in the blood. They're in this 1%, as are the waste products on their way out of the body. So like when we use amino acids for fuel instead of for building proteins, we have to get rid of the nitrogen they contain, and that nitrogen is transformed into a molecule called urea, and then urea goes to the kidney, and the kidney takes it out of the system, puts it in the urine, and we get rid of it that way. So in this 1% is also the organic wastes. So proteins, electrolytes, nutrients, and waste products are all in this plasma piece of blood. So 60%, 40%. All right, well, what about the formed elements? Okay, so all of these have a membrane around them. So rather than calling them cells, in order to include the platelets, we just call them formed elements instead. So platelets are little cell fragments. So they're not cells proper. They're the smallest thing you're going to see in a blood smear. And usually they just look like little dots. Up close, they look almost like shards of glass. You know, they clearly don't have a regular structure. And that's because they literally are little pieces of giant cells in the bone marrow that have sort of shed these pieces into the bloodstream. When you think of platelets, think of blood clotting, because that's their primary role. Platelets trigger the system that creates a blood clot and can seal a hole in a blood vessel, right? All right. Then the white blood cells. There are 5 different types, and we're going to talk about each of those here in a little while. So for now, we'll just call them the white blood cells. And then the red blood cells. Red blood cells are the most populous of all of the formed elements. In other words, of that 40% of formed elements here, almost all of that is red blood cells. And then there's a much smaller number of white blood cells and platelets present. All right. So since they're the most populous, we'll talk about them first. All right. So the red blood cell, I'm going to turn the light off here so you can see this a little better. As you've all learned in past classes, you know, it has this kind of flattened disc shape. It's a little like 2 frisbees that have been sort of stuck together upside down or with their flat surfaces together. The reason that the red blood cell is shaped like this is multifold. One, it increases the surface area. Inside the red blood cell, we find hemoglobin. Hemoglobin carries oxygen. In order for the oxygen to get out into the tissues where it can be used, it's got to pass through the cell membrane. So by being a flat disc, there's much more membrane, or there's much more surface area for the size of the cell than if the red blood cell were a sphere, right? So flat is better for that. Another reason that they're flat is so that they can stack up like poker chips. So you can see in this picture here, we have a couple of stacks of red blood cells. They're all sort of stacked on top of each other. That's called a rouleau. And when red blood cells are in that configuration, it's much easier for them to pass through very small vessels. So it improves the smoothness of blood flow by having this stack. And then finally, flexibility. By being sort of thin in the middle, and instead of a sphere but being flat, it means that the red blood cell can twist and turn a bit, particularly a young, healthy one. So, you know, when a red blood cell is nice and fresh, it can squeeze and bend and twist without breaking. And part of that is because of this shape that we have here. So as red blood cells get older, they start to lose that flexibility, and at some point they become too inflexible to actually continue in the cardiovascular system. Because what's interesting is many capillaries are actually smaller than a red blood cell. So the red blood cell has to squeeze to get through. It has to change its shape in order to pass through. All right, so we look. This is just a little movie of blood moving through the body. So it really does move. Red blood cells really do exist. And if you look at the small capillaries through a microscope, you see these patterns like that. So it's not, you know, I think sometimes we think of blood as moving sort of slowly through the body. It's not. When it gets to small vessels like this, it's moving along at a pretty good clip.
How big is it? That's not inside.
No, that's inside.
One person?
Yeah. It may not be a person, it may be an animal.
Those capillaries that we mentioned?
Mm-hmm. Capillaries, microvasculature we call it. In other words, capillaries, arterioles, venules, the tiniest little vessels, yeah. All right. So, red blood cells have really just one role, and that is to contain hemoglobin, and lots of it. So, red cells have lost their nuclei. They lose it in the bone marrow as they're being born, so to speak. The nuclei is removed. So essentially, the red blood cell is that disc shape, and all that's inside that disc is hemoglobin, tons and tons of it. So the way I think of red cells is they're bags of hemoglobin. They're crammed full of hemoglobin, and they have a shape. That's really about it. And the reason for that is hemoglobin is what accomplishes the red blood cell's primary job, and that is to carry oxygen around the body. So hemoglobin is this sort of complex-looking protein. It's actually made of 4 independent chains. So many proteins are one long chain that's been twisted into a shape. Hemoglobin has what we call a quaternary structure, which means that it is made of multiple parts. So there are 2 alpha chains, so alpha chain 1 and 2, and there are 2 beta chains, and they're all 4 are stuck together. So hemoglobin sort of has these 4 subunits. And each of these subunits has this disc-shaped molecule in its center called a heme molecule. The heme molecule is sort of where the magic happens. It's a very complex but very rigid and strong molecule. So this is what heme looks like. And you can see all these interlocking rings, right, of carbon and nitrogen atoms. That gives heme a lot of strength. And because it's such a strong molecule, it can actually hold on to an iron ion in the center. So because iron, it's a metal, right? It's a much, much larger ion than any of these others. If you look at the periodic table, iron is sort of further down in a different family. That means it's much, much larger. So the heme molecule holds on to this iron, and then this giant iron ion is what holds on to oxygen. So the heme has the iron holds the oxygen, and each protein subunit, so each alpha and beta chain, has a heme, which means that each molecule of hemoglobin can carry 4 oxygen molecules. These aren't oxygen atoms, so they're O2s. They're not O's, they're O2s. So it's the very same form of oxygen that's found in the air, and hemoglobin can carry 4 of those. So when we get to the respiratory chapter, we're going to talk all about the properties of hemoglobin and all about the properties of what we call gas transport, how oxygen is carried by hemoglobin, and then how CO2 is brought back. But for now, you can't really learn about red blood cells until you know what hemoglobin does and at least a little bit about it. So red blood cells are cell membranes chock full of these hemoglobin molecules whose primary role is to carry oxygen. All right. So all of the blood cells are constantly being replaced. One thing that makes living things, at least on Earth, fascinating is because they're always replacing their cells, in some ways, no part of you is as old as you are, except maybe your neurons, which don't replace. Red blood cells are no different. They only live about 120 days. So, if we're not going to run out of them, we have to keep making them. So they're born, they live, and then they die. And this diagram from your book sort of goes through that red blood cell life cycle, right? So we start here in the bone marrow. So it's in the bone marrow that red blood cells are born. And they're born from what we call stem cells. Now, they're not the same stem cells you hear about in the newspaper or whatever, but a stem cell process is simply, you start with a cell, right? That cell divides, right? We all know about that. That's mitosis, right? Cell division. What makes a stem cell is one cell then replaces the original, and another cell goes on to become something different. So that's all we mean by a stem cell pattern. And what this means is this can go on forever, right? Because this cell divided and was replaced, and then the second daughter cell went on to become something else. This can go and go and go and go and go because the original is always replaced, as well as creating an offspring that goes on to do something else. So this is happening for red blood cells and white blood cells in the bone marrow, and we just call that erythropoiesis, or the making of blood cells. And what we can see in that pattern is, in this last step here before it's released into the circulation, the nucleus is removed. So by the time cells appear in the blood, they usually don't have their nuclei anymore, unless there's been a big blood loss and the bone marrow is trying to make red blood cells as fast as it can. All right, so we get a new red blood cell out of that stem cell system. It lives about 120 days. So then what happens to it? Well, about 90% of red blood cells are destroyed on purpose. In other words, as they start to get older, they start to get less flexible, they start to be less pristine, so to speak. So in the liver and in the spleen, and to some extent in the bone marrow, they're snatched out of the circulation and destroyed on purpose, right? So that's most of them. 10% of them end up breaking in their lives. So like, as they get older, they can't quite fit through this particular capillary, and they break open. So 10% die in that way. So you will occasionally find little pieces and parts of red blood cells in the bloodstream because of that 10% that die out there. All right? So then what happens to them? Well, red cells contain some really important and valuable stuff. Most notably, iron. Iron is relatively hard to find in the environment. You have to, today it's as easy as going to buy an iron supplement at the grocery store. But, as we evolved, iron is relatively rare and hard to come by. So the body recycles iron, just like we do at the curb in some ways. So we want to get that iron out. So the red blood cells, the iron is removed, sent back to the bone marrow, and then that iron is then reintroduced into new red blood cells. So it's a true recycling system for iron. Well, what about the heme part of that molecule? I said that it's very, very tough and very strong. That's how it can hold on to that iron ion. Well, it's so strong that the body can't really break it down very well. So we have to have a system just for getting rid of the heme part of the hemoglobin. The hemoglobin molecule, it's all just amino acids, right? It's a standard protein. The body can get rid of proteins real easy. But this heme molecule is a different story. Once it's made, the body makes it. But once it's made, it can't really break it. So what it does is it transforms it into first biliverdin and then bilirubin. Bilirubin is a heme breakdown product. And it has a very greenish yellow color to it. Well, it turns out that bilirubin, the kidney, which is what gets rid of most of our waste products, it can't really get rid of bilirubin very well. So this heme molecule is a real pain. So what do we do with it? Well, the system that's developed is heme becomes bilirubin, then bilirubin is excreted into the digestive tract by the liver. So how do we get rid of broken down heme? Through the liver as bile. So the bile that goes into the small intestine contains bilirubin, and it's the principal way that we get rid of this byproduct of red blood cell turnover or red blood cell death. All right? So that's sort of where it ends, is with the final breakdown of bilirubin, or excretion of bilirubin. So we'll talk more about bile later, but this is the red blood cell life cycle, all right? All right. So you said that you talked about blood type in lab already, so this piece should go pretty quick. So, we name blood types by what we find on the surface, right? So type A blood has antigen A, type B blood, antigen B. Type AB has both. Don't get confused, there is no AB antigen. It's antigen A and antigen B, you have them both. And then type O has none of those antigens, okay? So that's what your red blood cells look like. Now the other piece of blood typing is, well, what antibodies do you have? For A and B, you have the opposite if you can. In other words, if you have antigen A, you're going to have antibodies to B, right? If you have antigen B, you're going to have antibodies to A. Well, what about AB? You can't really have antibodies to either, or you would have started out that dead, so to speak, right? So you can't have any antibodies. And then if you have type O, usually anyway, you have antigens to both B and A. So it's the antibodies are what determine what kind of blood a patient can receive, because it's the antibodies that are going to do the harm. So like if you give type A blood to a type B patient, these antibodies are going to stick to these antigens, the red blood cells are going to clump all together, and then the red blood cells are going to pop open and die. So you're going to have what we call a transfusion reaction. All right. The piece that's missing from here is the Rh factor. It's just another antigen that's found on the cell surface. So people who are Rh positive, they have an additional antigen. If they're Rh negative, they don't, right? And then they have the antibody sort of in reverse. All right. So if you have antibodies and antigens in the same patient or in a blood sample, this is what happens. The agglutination, or the cells all clump together. Once this occurs, the immune system attacks this like it's a foreign organism, so like it's a bacteria or a pathogen, and it just destroys these red blood cells. Now, what's the problem with that? Red blood cells contain a ton of hemoglobin, they contain a ton of iron. When you kill a whole bunch of red blood cells inside a patient, all that spills out, and it creates a massive immune response. So patients can die of all of this hemolysis, we call it. Hemolysis is just when cells break open. So we just have to be careful to match blood type, so we don't ever give blood with antigens to somebody with antibodies, because this is what ends up happening. And just like you should have done in lab, we can test blood typing using an antibody method, right? So if we put anti-A and anti-B and anti-Rh, we can tell what kind of blood type a patient has, which you guys have already done in lab, so we don't need to tell that, right? All right, so there is a different blood type problem that's sort of interesting, and that's called the hemolytic disease of the newborn, also called Rh incompatibility, right? You're going to hear about this in your careers because it is relatively common, particularly here in America, where we have a lot of genetic diversity. We've got genes here in America that have come from all over planet Earth. So we encounter blood type anomalies and differences more commonly than other nations do. Basically, what this comes about from, if a mom can be Rh negative, in other words, no Rh antigen, but she can conceive and bear a child that is Rh positive, right? Because the Rh gene can come from dad. So mom doesn't have an Rh gene, she's Rh negative. But dad did, so now the fetus is Rh positive. So what happens? Well, during the first pregnancy, nothing, because there's been no prior exposure. So at the placenta, mom's blood and baby's blood don't mix, okay? People think that they do, but they don't. Mom keeps her blood on her side, baby keeps baby's blood on baby's side. But antibodies do cross the placenta. So the blood cells don't cross or mix, but the antibodies mix from mom to baby and vice versa. All right, so in the first pregnancy, there's no problem, right? Mom's blood, baby's blood. Mom doesn't have antibodies yet because there's never been an exposure. Baby doesn't have antibodies to Rh because he's Rh positive, right? So he can't. He'd start off dead, as I said before. So at delivery, as the placenta separates, there's always some mixing of mom's blood and baby's blood. And this is where the problem comes. Because at that time, now baby's Rh positive blood is entering mom's circulation. And yes, mom's Rh negative blood is entering baby's circulation, but that piece doesn't matter, right? Because the baby doesn't have Rh antibodies, so it doesn't matter. So the problem comes after this. So now mom has been exposed to Rh positive red blood cells. What does she do? Well, her response is to make antibodies, right? It's a foreign cell. So the immune system is going to identify it as doesn't belong, and it's going to make antibodies to it. So now mom is producing anti-Rh antibodies, right? Okay, well, how does that matter? It matters for the second pregnancy, right? Because now mom has Rh antibodies, anti-Rh antibodies, and let's say she has another baby, it too is Rh positive, just like the last one. Very possible dad has the Rh positive gene, right? Well, now mom has antibodies to the baby's blood, right? And antibodies do cross the placenta. So you have this catastrophic result where mom's immune system is destroying baby's blood. So in the past, this was a baby killer because once you get enough antibodies and enough destruction of red blood cells, the fetus gets more and more anemic. It's essentially having a transfusion reaction all the time, and many babies used to die from this problem. And many more needed significant medical treatment, during or after delivery. Like what we call an exchange transfusion, where we take out all the baby's blood and we put new blood in because the blood has been so demolished by mom's antibodies. Now in this country, we don't see this very often anymore because we intervene at this step right here. So anytime a Rh negative mom gives birth to an Rh positive baby, before she goes home from the hospital, she gets an antibody injection, which prevents her from making Rh positive antibodies in the first place. It's called RhoGAM. It's given before discharge from the hospital, and then it's usually given again in a month afterwards. Yeah.
I actually have had my doctor's gave when I was pregnant,
Mm-hmm.
but my first shot they gave me when I was like 4 months old.
Yes, they become more and more aggressive
Yeah.
with it.
But then I had to get another one before we got there.
Yep. Yep. And the next, if you get pregnant again, you're probably going to have RhoGAM again. Because what RhoGAM does is any antibodies that are present, it's an anti-antibody. So it's an antibody that binds to an antibody and destroys it. So it's just an interesting, this hemolytic disease in the newborn, it's an interesting interplay of blood type and the placenta and how antibodies cross, but red blood cells don't. All right, so that's RhoGAM. Okay.
So if they don't catch it then, do they usually have a miscarriage? Is that like...
Many of the babies survive, but they're profoundly sick. When I was working in the neonatal intensive care unit, every year we'd have a few hemolytic disease in the newborn, and it's poor prenatal care. It's people who didn't get checked. Yeah. So like that exchange transfusion that I talked about, I've done a few of those where, you basically take the baby's blood out and you put new blood in to try to get their blood counts up because the problem is they're born with all these antibodies in their bloodstream. So you have to get rid of the blood in order to make it work. Yeah.
Is this the same as HLH, or is that just a completely different disease?
HLH.
Hemolytic, and you need a bone marrow transplant.
It's not the same thing as this.
It's
No.
not, okay.
No. I'm not familiar with it, but bone marrow transplant. Once you get the antibodies out of the baby's fine. It's not like their blood or bone marrow is screwed up somehow. Their blood is fine. Their problem is mom. Mom is attacking their blood cells with her antibodies. So, all right, let's do a question or two before we call today. Wow, that's went by fast. All right, formed elements make up about what percentage of blood? We got a little bit of a distribution here. And I know why. It's because that picture gives a broad range, right? That slide where it says plasma formed elements. Think 60-40. It can change. You know, like if you're dehydrated, for example, it's more like 50-50. If you're overhydrated, it could be 70-30. But for our purposes, think 60-40. So 45% D would be the correct answer. All right. Which of the following is not a component of plasma? All right, jump in there if you haven't. All right, I think that's everybody. All right, we got to split again. All right, which of the following is not a component of plasma? B, platelets, and that's because platelets are formed elements, and formed elements is blood is formed elements and plasma. So platelets are not part of the plasma, but plasma is that fluid component of the blood. All right, last one. Which plasma protein is a major contributor to the osmotic pressure of plasma? All right, jump in there because we're about out of class time. All right, good. Most of you found that answer or knew it already. That's D, albumin. Albumin, osmotic pressure go together. All right, we're out of time for today. Don't forget to get your homework in by midnight tonight. And I will see you all on Friday.
The number 6.